Welcome everyone out there in microbiome space. This morning we are going to cover some basic concepts on the microbiome inoculation, development, seeding, and dispersion. And so there's an article here that I want to review, and I will send it out in the email with the recording because it is very apropos for what we're trying to discuss here, and it's quite recent, and this is what we're finding out in so much of the science having to do with microbiome and probiotics, that a lot of companies are using antiquated old research, and this is what's very different about microbiome labs, is really trying to stay up on the most advanced research. So we see this quote here. This is an article that's taken from the year 1684, and this was published in Philosophical Transitions, and it was breaking news. Though my teeth are quite usually very clean, nevertheless, when I view them in a magnifying glass, I find growing between them a little white matter as thick as wetted flour. So this was the discovery that we are not alone, that there are organisms everywhere. And this article is talking about the different technologies and how 16 RNA gene sequencing is one. It's the older one. The total DNA sequencing or shotgun metagenomics, mass spectrometry are the up and coming. And what I wanted to get to, you see a lot of good information covered here about the gastrointestinal microbiota, is here and how the microbiome, the many microbiomes of the body develop their own unique environment. And here this is talking about hair, nostrils, skin, vagina, oral cavity, esophagus, the stomach, the colon. And then it's discussing here the different organisms that make up those different environments. And this is showing the snapshot, the human microbiome. So now we're just looking at how throughout the world different microbiomes will change and how that is unique for location. And here we're looking at different associations with disease, diet, and the microbiome host. And what I wanted to get to, and this is talking about the different short chain fatty acids and their role in the body and populating the human gut. And here we're talking about 1 to 2 years of life resembling the final adult pattern. And this is where we're talking about that 835 days, and after which the microbiome is mature. So this is just good documentation. Here's showing that the factors influencing the gut microbiome development are diet, the womb, mode of delivery, gestational age, genetics, environment, and antibiotics. And here showing healthy development versus disease development, and then how the populations will change in a diseased environment. Okay, and then they're looking at how do probiotics work, and this is a very important statement here. You know, this study was done in 2016, and they're saying, while there is some evidence from previous reviews that probiotic interventions may benefit those with disease-associated imbalances of the gut microbiota, there is little evidence of an effect in the healthy individuals. And this is when they began to realize that unprotected or non-spore bacteria really weren't doing that much as far as probiotics. So there's some research there. We'll talk more about the TMAO in our presentation next week when we're talking about some of the updates in the new BiomeFX kit. Okay, I think it's Clostridium. Okay, and this is discussing the American Human Microbiome Project, the European Meta-Human Intestinal Tests. So it goes, deeper into the technology and, the metabolomics, and then it gives the summary here. So I wanted to tell you I'm sending this article out in an email so that you can have that reference. And then I had sent out just a thank you for those who have registered for the June meeting and some terms. And one of the terms was seeding. And, there's been some, 4R programs out there in the past that alluded to you can reseed a microbiome with probiotics. We know that is false. We know that the seeding, the initial inoculum is primarily from the mom, the environment, the breast milk, and that's mature after 838 days. And this dispersal that we're talking about is that, even though the initial inoculum is, through the vagina, that after time, each of these environments form their own unique microbiota and microbiome. These original organisms create an environment that becomes conducive for other organisms to join them. But that window of opportunity for joining the club ends at 838 days. And there is no more reseeding. You can only influence. So that is kind of the important fundamental concept to understand. And that helps us really understand why the probiotics, the lactobacillus and the bifidus, the non-spore probiotics have really failed in functional medicine. And now I am going to end this recording so that I can start one